Okay, so very quickly, we're going to be talking about B cell development. One thing you have to know about B cell development is that approximately 55 billion B cells per day die in the bone marrow. Okay, that's a pretty interesting number. Now, like I just said, where does B cell development occur? It occurs in the bone marrow. Okay, now we have this hematopoietic stem cell that gives rise to some sort of common lymphoid progenitor. And this common lymphoid progenitor will go ahead and become committed to either a pro B cell lineage or a pro T cell lineage. In this case, we're talking about a pro B cell. Okay, we have a whole other video talking about a pro T cell. But the pro B cell, when it becomes committed, what does it do? Afterwards, it can become a large pre B cell or a small pre B cell. Okay, there's different stages of a pre B cell. So you can go to a large pre B. In the large pre B, this is your first checkpoint. This is important. What do we have in the large pre B? In the large pre B, we have V to DJ recombination. Okay, so in the pro B cell, we already did our D to J recombination. And now within the large pre B cell, we're doing our V to DJ recombination. And after that's done, we're going to express that heavy chain. Okay, we're going to express that heavy chain, the immunoglobulin heavy chain. Once we do that, we've also made some surrogate light chain proteins. These guys are invariable, okay, meaning they're the same in everybody. So these surrogate light chains, you make them on the side, and this is to really test to see if the heavy chain that you've made is even responsive to the light chain that you're going to make in the future, right? And of course, at this stage, you have your immunoglobulin alpha and immunoglobulin beta signal transduction proteins, which are going to carry that signal down, okay? Just to see if it works. Now, the first checkpoint is this. If you cannot make a pre-BCR, okay, so if you cannot make a pre-B cell receptor, especially that heavy chain, or if you can make it, however, it doesn't work, okay, it doesn't work, then that can lead to apoptosis, okay, that can lead to apoptosis. Okay, let's move on. So now we're in the small pre-B, okay, you can clearly see this, the cells become smaller. So in the small pre-B, what do we have here? In the small pre-B, we can have V to J recombination. Why V to J? Because it's a light chain now, okay? We tested our heavy chain works. Now we need to finish making our light chain. And you can see here, this is our light chain recombination occurring here. Okay, so V to J recombination. And now that protein that we had on the surface comes back in here, and now we have an intracellular mu protein. Okay, intracellular mu protein. Very important to remember that we always make immunoglobulin M first, or Ig mu. Okay, that constant mu always has to be made first. Then we can move on to expressing other ones like delta, okay, or IgD. Now, another thing when it comes to the light chain, you always, during V to J recombination of the light chain, you always try the kappa locus first. If the kappa locus fails, okay, and you've tried all of it, then you can move on to make to using the lambda chain, okay. That lambda isotype, okay. That's all I want you to know. Okay, now let's move on. After the small pre-B, okay. Actually, before we do that, the small pre-B is your second checkpoint. Okay, so the first checkpoint was large pre-B. Second checkpoint is your small pre-B. In the small pre-B, if you cannot make a B cell receptor, okay, so if you can't combine the light chain and the immunoglobulin heavy chain together and make a functioning pre-B cell receptor, you're going to go through apoptosis. Okay, so your second checkpoint is if you cannot make a B cell receptor. Okay, no B cell receptor complex. That's that. Now let's say you did that though. Let's say you made that. Now look at this. The immature B cell has what? It has a fully functioning immunoglobulin M on it. Okay, and in this stage, the immature B cell is going to go through selection. Okay, it's going to go through positive and negative selection. Negative selection. Let's talk about this. So this is positive selection here. Within positive selection, if you have a weak recognition, of what? Of these self-antigens on the surface of these stromal cells, or the hematopoietic stem cells, okay, or some of these macromolecules, okay, that are within this region. If you have a very weak recognition of those antigens or no recognition, you're good to go. Okay, you'll turn into a mature but naive B cell, and you'll start expressing immunoglobulin D on the surface as well. Okay, and then you'll go out into the blood. That's positive selection. You can also have a negative selection of this immature B cell, and I'll explain to you how that works. The immature B cell, if it has a very strong recognition of this, it could be the stromal cell, like I said, or a hematopoietic stem cell, or it could be a macromolecule, right? One of these. If it recognizes these antigens extremely strongly and binds, okay, that's not good. That's autoreactive. We don't want that. Okay. What I want you to take note of is it can actually be given a second chance. It can go through light chain editing, light chain rearrangement. Okay. This gets tricky because people will start asking, well, why can't we re-edit the heavy chain? And that's because you've already made the heavy chain. Okay, the heavy chain has already been expressed, one, and two, you've already gotten rid of all of your D segments. Okay, so if you were to kind of imagine here for a second, take this for example. Okay, imagine I had a gene segment. I had V. Okay, this marker doesn't work. Imagine I had a gene segment. I had V1, V2, V3, and then I had D5, D6, and I had J1, J4. What do we do normally? Normally, when we're making the heavy chain, we do D to J first. Okay, so we do D to J. Let's just say I pick D6. Okay, I took D6 and J4. Okay, so now I have D6 and J4. Okay, and let's just pretend after D6 I had D7, D8, D9, D10. Everything past this D6, everything in between these guys, okay, so everything in between these guys was eliminated. Okay, so everything in between those guys was eliminated. So J1 would die, D7 to like D10 would die. Okay, so now we have just these guys. And then what else do we have? Now we gotta do V to DJ. Okay, so V to DJ. From V to DJ, I had V1, V2, V3, and then I had D1, D2, D3, D4, D5. Let's say I chose to do V2, whatever, okay? So now I do V2 with these guys. Look at what's gonna die. V3, D1, D2, D3, D4, D5, D6. We might still have some V's and some J's. However, all of my D segments are gone. If all of my D segments are gone, am I gonna be able to reopen that locus and then start heavy chain rearrangement again? No, I can't do that. Okay, I can't do that. So remember, that's the reason why we can't do that. And you might be asking, okay, well, I get that, but we have two chromosomes. Why can't I just open the other chromosome? Because during this heavy chain rearrangement, we already went through allelic exclusion. Okay, we successfully made a working heavy chain, and so we excluded the other allele. We can't go back and do reopen it. Okay, we can't do that. But within here, we get a couple chances because the light chain. Remember, we had V to J, right? So we have a lot of segments to go through. Okay, no matter which one we chose, right? So it could have been V1 to V100, and then J1 to like J60, and we have a lot of options to go through. So we can edit our receptor as many times as we want. Okay, and this is where it gets better because we know we have two isotypes of the light chain. We have the kappa and then the lambda. Okay, so if kappa fails, I can go to lambda. Okay, so let's say I exhausted all of my VJ recombinations within kappa. If that fails, and I still haven't able to make a receptor that is not strongly recognizing this, then I can go ahead and I can make I can go ahead to the other locus and use the lambda. Okay, so I can use the lambda. Okay, and again, remember, allelic exclusion does play a factor here as well. Okay, so you can't, if you make a successful one, you can't reopen the other one. However, we have not made a successful one here, so we can keep reopening the other allele as well and try it out. But we have to exhaust the this chromosome, this allele's chromosome, the kappa and the lambda isotypes in this one, then we go on to the other one and then try it out if we still haven't. So if you're not getting it by now, you can do a billion different edits, okay? And if one of them works, then you can go back to positive selection and then you can become mature. If you exhaust all of your VTJ recombinations, whether it's on the kappa and lambda isotypes on the first or second allele, you're going to go through apoptosis, okay? Because you know what? The cell's like, okay, you know, I tried to give you all of these different chances, but, you know, maybe there's just something wrong with the heavy chain that you just can't fix, right? It's not the light chain that's the problem. And so the body will eventually just kill this one, okay? And that's what you need to know. All right. But again, this portion was called negative selection. If we go through positive selection, we become a mature, naive B cell, we move out to the blood, and we express IgD. Okay? Now, when recepting editing fails, okay, when it's failed, it's called clonal deletion. Okay? Remember that. It's called clonal deletion. Okay, and that's pretty much everything I want you to know about this.